Good afternoon, my dear students. We have discussed in the previous class regarding the expression of gene. Expression of gene. Genes are there, which are the hereditary repositories of various traits of an individual or an organism. And all these genes are expressed. To have a gene merely is not sufficient. Its control of expression is also mandatory, is a must. Otherwise, there is going to be disease or death. So we have learned thoroughly in the previous class. There are two different kinds of genes existing. One are constitutive genes. All the time they are expressed, and another one are repressed or induced. They are inducible. If some signal is there, some substance is there, some material is there, then that particular genes start expressed, and they participate in the metabolism of that particular substance. That is, the substance is called inducing substance or induction substance, and the signals are inducible signals. And we have studied a model which is called as lactose operon model, lac operon model. This is mainly deciphered or mainly discovered by Jacob and Monod, two scientists from French origin. Along with that, contributions of Andrelov and Severo Ohava are commendable. And in this model, we have learned not only the gene which is producing the protein, and its controls are in the upstream. They are called left side. They are called upstream. Upstream controls are also we studied. The structural genes, that is the protein supposed to be expressed, synthesized. There are three in the lac operon concept. There are three genes: LAC-Z, LAC-Y, and LAC-A. And all the three genes in total are controlled by controlling areas. One is an operon area, another is a promoter area, and there is a mini gene, a LAC inhibitor, or this is an inhibitor producing a repressor molecule, a repressor protein. And this repressor protein always breaks these three genes not to be transcripted by RNA polymerase, which is being adhered to the promoter zone, promoter area containing RNA polymerase. Operator area is either allows or stops the polymerase enzyme to go forward and transcripts these three genes. And this control is exerted by this LAC inhibitor, or this particular gene that is being producing a repressor molecule, breaking molecule. This goes and joins here, and when it is joined in this condition, the repressor molecule joined in the operator area, then polymerase cannot go. This is something like a big wall that is being created here. Whenever the inducer material, that is the lactose, when a lactose is there, these three genes should produce proteins which are enzymes: beta-galactosidase, perimidase, and transacetylase. All these three enzymes put together digest or metabolizes lactose to produce energy to the organism. The organism is in the E. coli. The model we have taken as an E. coli model. The other operon concepts also we have mentioned, and those concepts are tryptophan operon, galactose operon, trip operon, gal operon, and histidine operons. So this is how the regulator gene, promoter area, and operator area, RNA polymerase, and from the regulator, that is the inhibitor protein gene producing the repressor molecule. Repressor molecule ultimately all the time is joined to the operator area, and then producing a wall, and RNA polymerase cannot transcribe, ultimately no protein. So now, whenever the material is available, that is the material allolactose or lactose, when it is available, it will bind immediately to the repressor molecule and changes its three-dimensional orientation. No more, it can bind to operator area. So all those ones which are being stopping the signal are being removed from this particular area after joining by the lactose or lactose inducer molecules, and that is how RNA polymerase is freed, and all these genes are transcribed and producing these three proteins. Ultimately, they will digest, metabolize the lactose. So this is how there is a control, nice control. Whenever the material is there, the proteins are rather the genes are transcribed, proteins are produced. So then, after learning thoroughly about this, now we shall enter into another very, very important and fascinating topic, DNA recombinant technology. DNA recombination, combining and recombining. Why is this said recombinant DNA or DNA recombinant? Why they are called it? Because from two different organisms, say a human gene and a E. coli DNA material, when they are joined together, that is recombination. So two different species DNA materials are joining together is called as a recombinant DNA. So how we can define the method by which a living organism or its parts are used to change or to incorporate a particular character to another living organism. This is now called as a DNA recombinant technology or recombinant DNA technology, or in a popular word, genetic engineering. So these works are all, or otherwise these methods are all commonly called as genetic engineering. What is the biggest advantage of having this particular one? To produce quantitatively so many biomolecules. My dear students, suppose we know a disease, a very, very definitely a serious disease, which has been affecting the whole world's population since ages, unknown since centuries. That is the diabetes mellitus. What is diabetes mellitus? It is the absence or not or resistance to the hormone insulin. Let us say first absence, lack of insulin synthesis in the individual is ultimately produces diabetes mellitus. So what is the treatment? We need to give insulin. Where from we get insulin? We cannot chemically cannot synthesize it, like various aspirin, etc., paracetamol, etc., which chemical molecules, from the chemical molecules we can synthesize in the pharmaceutical industry. But now insulin in the pharmacy, how do they obtain? In the starting, when they have started treatment after discovering that diabetes mellitus is due to the lack of insulin, and that is due to the destruction of islet cells, beta cells of Langerhans cells, and they are not able to produce the insulin due to disease or death, and ultimately producing the diabetes mellitus. Then the treatment is obviously injecting insulin regulatedly. And insulin, where do we get? We'll get only in the pancreas. So in the initial stages, the treatment of diabetes mellitus is by way of extracting insulin from the pancreatic tissue of different animals. And the most popular one is a pig, and hence they used to call it as porcine insulin. And another source they have taken as bull's ox. So bovine insulin. So two different kinds of insulins they used to prepare. So what is needed now? So many animals, pigs and ox, they have to be sacrificed, and their pancreases have to be taken out. And from there, we need to isolate the insulin and pack the insulin in the vials, and then they are distributed among the population in the pharmacy. So now, so many animals has to be killed. At least insulin, in the case of insulin, we are somehow able to manage. Imagine the example of growth hormone. Where do you get the growth hormone? Pituitary. Unfortunately, the pituitary from the animal sources are least capable of doing the effect or eliciting the effect on human beings. And hence, cadaveric pituitaries. So, okay, so human pituitaries need to be harvested. That is the ideal one. So that is made the growth hormone distribution is not possible, because how to get the human pituitaries? Not only that, the first one. Second is from about 1,000 pituitaries, if you can get it, 1,000 human pituitaries, you can get it. 1 microgram of growth hormone, you will get it. So what would be the cost? And is it really practicable to practice? So in the short stature, due to growth hormone deficiency, to give them growth hormone is such a difficult task, because producing growth hormone is not that easy. So now, after having the DNA recombinant technology has come out, we are able to produce this insulin and growth hormone in highly purified. Not only producing, we are producing in a very highly purified manner. When we are extracting from the organs like pancreas or pituitary, we are mincing them, we are crushing them. While doing so, we are also able to have a lot of contamination of that particular animal proteins. Not only that, if they are suffering from some diseases like hepatitis B or any HIV, then there is a very likelihood this material enters into this growth hormone or insulin preparations. And hence, there is a danger of giving a disease along with this treatment. So these two things have been overcome with the help of this DNA recombinant technology. Then, that is one very, very important use. And the second use is specific probes for diagnosis of diseases. So if you want to diagnose a disease, and for that purpose, you need certain probes. And these probes are anti to that protein molecules which are present in those diseases. These can be produced by recombinant DNA technique. And then, antenatal diagnosis of genetic diseases purposes. Antenatal diagnosis of genetic diseases purposes DNA recombinant technology is useful. One is cystic fibrosis. Examples, diseases which can be identified. Cystic fibrosis. There is one disease called as cystic fibrosis. Second one, phenylketonuria. This is an inborn error of metabolism due to phenylalanine hydroxylase enzyme is deficient in the humans. And then another purpose is to identify viral particles or bacterial DNAs if they are present in the cells of the individuals. And then you can demonstrate viral integration into the host. So if viruses are integrated into the host, that is, they have entered into the cells, can be identified by this technology. So you can identify the viruses in the cells, say for example lymphocytes. Again, you can detect activation of oncogenes in cancer cells. Detecting activation of oncogenes. Oncogenes are responsible. They are best present in the They are in the cancer cells. In the cancer cells, you can see the activation of oncogenes. Oncogenes does present in the cells normally, but when they are activated, then that particular cell is going into the biology of cancer. So that means that cell may turn to cancer. So this is another use from recombinant DNA. And you can pinpoint location of a gene with this technology. You can pinpoint the location of a gene in which chromosome. Suppose a chromosome is known, in which part of it, in which locus of the chromosome it is present, can be identified by this recombinant DNA technology. And then it will be useful also in the gene therapy. This methodology is used up and can be used in the gene therapy. Well, so these are all the uses of recombinant DNA technology. It has revolutionized our medicine, especially treatment and diagnosis. Well, to enter into the DNA recombinant technology, the first inventions are the invention of endonucleases. The bacteria are having certain nucleases which can cut the DNA of these bacteria. They are being observed initially by Werner Harper. And they have a specific nature of cutting that is first observed by Werner Harper. And Smith has identified or isolated these enzymes. And Daniel Nathan has said they are restriction endonucleases. Always, whenever they are cutting in a specific form in the DNA. They are cutting the DNA in a specific form at a particular point, always at that particular point. This is more important. So, Harper, Smith, and Daniel Nathans, all three, has given the first important step to design this DNA recombinant technology. And hence they got Nobel Prize. And then Palberg has entered. And then the types of various restriction enzymes. Now, various restriction enzymes. Note down the E. coli, it is called as, one is called as E. coli. That is E. coli restriction enzyme I is 1. That is E. coli RY13, its proper name. And another one is HIND3. This is another restriction endonuclease. Restriction endonucleases are very, very important. And the first process in the DNA recombinant technology is the genetic material is being subjected to restriction endonuclease attack. Restriction endonuclease attack. The first step is subjecting the DNA to the restriction endonuclease attack. Wherever the gene is there, say for example, insulin gene is there, that particular gene can be isolated. That can be isolated after this cutting. So the large DNA is going to be cut into pieces, and that they are specified pieces, and the edges are specially cut. They are called as sticky cuts. Stick, adhere. They can adhere. So that is the main character of restriction. Another restriction is the TAC, Thermus aquaticus 1. And then another one is HPAL, Haemophilus parainfluenza. So these are all the restriction endonucleases. Restriction endonucleases is always a short notes. So now, after having the human DNA or RNA, say, with the help of restriction endonuclease, you can have a piece of DNA. Suppose if you are not coming from the other side, you have another way. A protein is there. Correspondingly, the human mRNAs will be there. For every protein, before that step, mRNAs will be there. And depending on the mRNA, the reverse transcriptase enzymes, we have also learned about it. Basing on this, they can produce one strand of DNA. That is the code, the template strand can be synthesized. The coding strand can be, no, with the help of mRNA, you can get the template strand. Basing on the template strand, the coding strand also can be later on synthesized. Initially, the RNA-dependent DNA synthase, that is the reverse transcriptase. After that, DNA-dependent DNA synthase, that is the coding strand. So both these strands. So from the protein, you got the mRNA, or directly you get the mRNA, and this mRNA, you can synthesize a DNA, the gene corresponding to it. What is this gene? Let us take it. Granted, insulin gene. So human insulin gene is being there with us. So how to, using this gene, producing the protein of insulin? Now we need to introduce this particular gene into the experimental animal. Our experimental animal now here is micro animal, that is E. coli. So how to put this one into it? There are certain microorganisms which can enter into the E. coli, subbacterial organisms, and they are one among them plasmids and cosmids, and one mainly is plasmids. Then plasmids are normally present in the bacteria. They confer resistance to the antibiotics. When these plasmids could be or can be isolated, taken out from certain bacteria. And once these plasmids are there, and this gene containing insulin is there, both of them, again with the help of restriction endonucleases, you can join this gene onto the plasmid. Plasmid is originally from bacterial source, and this plasmid is cut at two different parts with the help of restriction endonuclease. And the big large DNA of human is also cut by endonuclease, restriction nuclease. And that insulin gene is able to be identified. And this gene and this cut plasmid, both of them are made to interact together. And what will happen is this gene of insulin is joined in this plasmid. So this plasmid of the bacterial origin and the gene of human origin are now in association. This is cloned DNA. This is a cloned DNA. plasmid vector cleaved by specific restriction endonucleases. Vector is carrying human gene. This is been synthesized. Clone DNA is being incorporated into it. And now this whole thing is being now allowed. So now one character in this. One character in this. What is that character? This is human and this is bacterial origin. And suppose a human being is having a lion's head, or a human being is having elephant's head. We do have in our mythology Lord Narasimha and Vinayaka. So they are human body with animal heads. What are they? One is lion, another is elephant. Similarly, in the Greek mythology, they have such gods. And they said chimeras. That morphology is called as chimera morphology. And hence, this is also called as chimera. This is called as chimera. And then this plasmids with the DNA, cloned DNA, is being allowed to react with the E. coli. So now this and this interact, and then this plasmids having capability to enter into breaking the wall of E. coli and entering into the cytoplasm of the E. coli. And then here now they try to start expressing completely. Along with that, this gene also expressed. And that certain E. coli are able to express certain of them, the effort will be wasted, or somehow the reaction between these two is not fruitful, not able to enter into the E. coli. And such E. coli are being removed, and those functioning E. coli are being selected and further grown. So this is what is called as transfection into host bacteria. The plasmid is able to enter into the bacteria. This is called transfection. And then selection of that bacteria carrying the human gene that is having an antibiotic sensitivity. And then the bacteria are being very much amplified in large quantities. And when they are grown, they are producing all the proteins which is present in the plasmid. Along with that, the insulin. And the insulin is being separated by a technique chromatography. And that is being concentrated, purified, and then insulin, recombinant insulin is recognized. Or in fact, any recombinant protein. So what are the steps here? Here, human mRNA or any DNA, RNA from the other sources, and a cloning can occur. That is, it is cut by restriction into nucleases, and then that one is made to associate with the plasmids, and this is plasmid vector cleaved by specific restriction endonuclease and a vector carrying human genes, and these were transfected into the host bacteria. And host bacteria are able to divide and they grow. And once they grow, some of them they are having functional human gene, and they are cloned. That means the functional ones are all being cloned and produced in large concentrations. And they are grown again and again, and then producing the proteins in them. And those proteins are all being purified, separated, and then we can get the insulin. This is the recombinant insulin, or recombinant protein, and that protein can be used for other purposes. Now, what is called vectors? Which is the vector? The plasmid is called as vector. So, plasmids, cosmids, and various other ones can be used as vectors. They are circular, double-stranded DNA molecules normally present inside the bacteria, and they confer antibiotic resistance to various microorganisms. So now here, in this whole technology again, we will review it. What are the various steps to be done? Preparation of specific human genome or a cloned gene. And then preparation of chimeric DNA. That is chimera. That's just now I told you. So chimera, a part of human gene, remaining is a bacterial plasmid gene. And then the cloning of here, which one is functional, is not functional. Cloning of chimeric DNA. A clone is a large population of identical bacteria or cells that arise from a common ancestor molecules. This is also, we also had cloning of a cell. When a cell which is being taken from any organism or any animal, say about a sheep, and then that particular cell is already in the completely it has gone into a specified type of cells, muscle cells or fibrocytes or heart muscles. So they are specialized. But those cells, some cells can be taken. On them, there are some factors, transcription factors and growth factors are being added. And once they are added, it will become again going back in the differentiation into specialized cell, and they'll go back to pluripotent cell. And that is what is the and from there an animal can be again it can be developed. This is what is called as cloning, another technology, cloning. And that is with that technology certain animals are being cloned, that is without having this male and female union, and then sperm and the ovum meeting is not there. Without that, from the organism's cells, pluripotent cells are being produced, and from there again you can produce the organism that is producing another sheep, a dolly. In the literature we have the first cloned one, sheep. Then this is what is cloning, so another step. And then transfection of vectors into the host, and expression of vectors, and later on the human recombinant proteins are being isolated. In this way, what are the various fruits we have got? Now we are able to produce the insulin for the sake of treatment of diabetes mellitus. We are able to synthesize the growth hormone, which was which is very highly Herculean task, because gathering about 1,000 pituitaries from various sources and then crushing them and isolating them from there, isolating the growth hormone and preparing the growth hormone. So Herculean task became reasonably easier task with this DNA recombinant technology. And then the very important one, another one, is FH, follicular stimulating hormone. We are able to produce follicular stimulating hormone, and its fruits we are enjoying. That is in the test tube technique, you say. In this test tube baby technique, you need to harvest ovum from the mother, because there is a problem in the development of the ovum in the mother itself. And then to develop the ovum, you need these follicular stimulating hormones, a very high potent and purified ones we are getting from the recombinant DNA technology. And it is IVF technique. Now we need FSH. This is one. And secondly, clotting factors in the hemophilia, the disorders where clotting mechanism is altered, and then whenever there is a trivial injury, the individual may go bleed. So these diseases to control, you need clotting factors like 9, that is hemophilic factor, Stewart power factor, something like that. And then there are what are called TPA, plasma angiotensin, is another one. And then hirudin is another protein. And these proteins, which are used in the clotting mechanism to activate it or to reduce it, these can be mediated with the help of these proteins. Next is another more very, very popular one, where all gets the vaccination against hepatitis B. Hepatitis B is a virus. This virus attacks the liver of a healthy person. Once it attacks, it produces jaundice. You all must be knowing about it. So hepatitis produced by infective hepatitis virus. Infective hepatitis virus. And there are types: A type, B type, C type, D type, etc. And B type is mainly transmitted by the blood products, and hence all those health personnel will be taking vaccination against B. This protein is also genetically engineered and hepatitis B vaccine. And HPV, hepatitis, that is herpes virus. And these viruses, vaccinations, recombinant DNA technology fruits. And now we have growth factors and cytokines. And they also are being synthesized by, like interferon, interleukins, they are all being synthesized by recombinant DNA technology. And for the treatment of cancer, certain monoclonal antibodies are being produced. These proteins are very, very, very specific, and hence they are called as monoclones. Very highly specific ones, and monoclonal antibodies. Something like rituximab and trastuzumab. They are the two. Rituximab, trastuzumab are two monoclonal antibodies which are being used in the cancer treatment. And recombinant enzymes for certain investigative techniques, in the methods, certain enzymes are needed, and those enzymes are also being synthesized by recombinant DNA technology. And recombinant HIV proteins, mainly for HIV test kits. HIV, whether a person is having it or not, we do in the laboratory tests. We take a small drop of the individual's blood, and we will place it onto the testing device, and this testing device will give a signal, a red signal, red line, a red dot, and this is how positive or negative can be identified. And for this devices to be synthesized, you need HIV proteins, GP proteins, or protease proteins, etc. Envelope proteins. They are there in the HIV molecule. And then HIV virus, rather. And the HIV viruses are being disintegrated, and these proteins are being synthesized in the recombinant technology also. And they are used for the testing devices of HIV, etc. in the ELISA procedures. And then various crops, especially in the farming, that is the rice or wheat plants, they should be resistant to insecticides or herbicides. And herbicide and insecticide resistant type of crops designing requires DNA recombinant technology. And then various proteins, say for example, suppose albumin concentration is very low in nephrotic syndrome of an individual. And if albumin is not there, there is going to be edema. There is going to be change in the oncotic pressure of the blood of that particular individual. And hence, it is the best treatment to infuse some amount of albumin. So this albumin synthesis can be done by recombinant DNA technology. Or there is another important protein, bone morphogenic protein, BMP. For the sake of wound healings, they both BMP, or for certain test procedures purposes, BMP is being synthesized. And fibrinolytic and thrombolytic agents are also synthesized from DNA recombinant technology. And lastly, we shall discuss a little bit about the Human Genome Project. Human Genome Project. Genome. We know insulin, so many amino acids are there, A chain is there, B chain is there, inter-peptide linkages are there, intrapeptide linkages are there. All these things we have learned it. And then what are the amino acids 1, 2, 3, 4, 5? And to know about them, we know it. And then if that is only few hundreds are there. But here our genome is having 2 sets of chromosomes. Each set of chromosomes, that is 23, if you take the completely stretch the 23 DNA chromosomes, DNA molecules in a single stretch, they will contain about 3 billion nucleotide pairs. 3 billion nucleotide pairs. Out of them, about only 10,000 genes are there. That is hardly, it will be about 2 to 3% of the total genome. This whole stretch of deciphering 1 to that the 3 billions and odd number of nucleotides 1, 2, 3, 4, 5 without having any mistake in it, deciphering them, knowing about them is otherwise called as a genome project. In this genome project, we not only know about this the nucleotide sequences, but also various zones, pieces, what is their functions, deciphering the functions, the whole thing. Otherwise, the breed, the genetic breed of an individual, the DNA fingerprints completely to know about them. This process, the whole process was initiated in 1990. And the team leader is, who is the inventor of DNA? Watson and Crick, or rather Crick and Watson. Crick is the senior, Watson is the junior. And in 1990, Watson became the leader of this particular project. They said this is Human Genome Project. It is mainly controlled from the USA under the leadership of Watson. But other leading countries like Britain, Germany, Japan are also, France are also participated in this genome project. One chromosome, one particular country, or 2, 3 chromosomes in a particular area, they have distributed among themselves and completely deciphered. And this process continued for 10 years nearly. And finally, they could able to decipher the nucleotide sequences in all the projects together, all the places together, and come to a conclusion in 20,000. And then onwards, one by one, what are their functions they are going on? Fingerprinting methodologies they are establishing. So one by one, this Human Genome Project is taken shape and giving fruits. And finally, ultimately, we have the book of human life in it. And this particular deciphering and knowing about them, each chromosome, what is its functions, what is its primary conformation, what is the secondary level, and etc. And finally, we have 23 chapters in the book of human life, and that is the human genome. Human genome. And then the last is, with having all these things, knowing about it, what is our ultimate aim? We want to treat. That is gene therapy. Gene therapy. They people are attempting, after knowing a particular gene is at a particular place, well, that particular gene can be isolated. Say, for example, insulin gene we know. It is there on that particular chromosome at that particular loci. And there, using the restriction in the nucleases, we cut open that particular gene this way and that way, not exact gene, some long stretch of that particular gene, that area, polynucleotide area, is being cut and then is being incorporated either into the plasmid or into the cosmid. And now, plasmid cosmid is and to put it into the chromosomes, put it into the bacteria. But now, where do we want to put it? We want to put it into the human being. How can we put it into the human being? The best, what we have done, you see, we are make use of adenoviruses, which will produce common cold. Understand? Adenoviruses. Some few retroviruses, which are innocent retroviruses. And these viruses are being taken, and again using the endonucleases action, this gene is incorporated in that viruses. So common cold producing virus, into that the insulin gene is being incorporated, or cystic fibrosis gene is incorporated, mainly cystic fibrosis gene, because cystic fibrosis mainly affects the lungs. And then this adenoviruses will be installed into the lungs. Adenoviruses having, because they are common cold producing viruses, so they can happily live in the lungs. While living there, they can also produce its own proteins. Along with that, the insulin gene, which is hiding in them, we have introduced it deliberately into it, and that also will be expressed out. That means that gene also will be expressed, and insulin will be produced. Some amount of continuous insulin will be produced in the pneumocytes, or through pneumocytes they may creep out into the other cells, few other cells, and there also it will produce. So this is how we want to exploit the viruses. But what has happened, when we are exploiting the viruses, they also know to exploit the human beings. And the deadly thing has happened now. So obviously people has experimented with these viruses. And either they might have newly arisen, or they have taken virulence or somehow, and at times these are certain bad ill effects of the fruits which we may get through adenoviruses. We may get insulin, and we may treat the diabetes mellitus or cystic fibrosis or Alzheimer's disease, and severe combined immunodeficiency syndrome. So we are, mind you, my dear friends, here we are deliberately taking the viruses, which are innocent viruses, which will not produce major ill effects onto the human being, and they are very, very, very timid ones, and they are cut open into them, into their heart. Otherwise, only colloquially I am saying that there is no heart in the virus. Into them, we introduce the required gene, human gene, into it, and we grow in large quantities, and they are prepared as insulators into the nose, and then they grow production of chimeric DNA molecule by using E. coli restriction in the nucleus. Red arrow show the site of cut by E. coli. And then this is about the recombinant DNA technology. There is a various, you see the sticky patches. This is a gene, this is a plasmid, then this is a sticky patch, the gene, and they can cut open. This is a double helix. Then it can rejoin back. The reason for rejoining back is they have a specific sequences. While you speak a word from forward to backward, backward to forward, like you say Vikatakavi, similarly a madam. The other side is also madam. So such a cut it does, and hence they are able to adhere the plasmid end is going to adhere the gene end. Similarly, the gene end is going to adhere to the plasmid end, where the restriction of the nuclease has cut into it. Both these sides will have a joining capability. So this is how the DNA recombinant technology is useful in the human medical knowledge, medical medicine to be. It is of great use to treat various diseases in the medical science. Thank you very much. Hope you could understand this DNA recombinant technology. Thank you.